Hello everyone and welcome back for chapter 18. So let's get through this as quick as we can. So sensory receptors, the big division here is whether it's something that tells you what's going on outside your body or inside the body. So interoceptors are things that tell you what's going on in the body. So you have blood pressure, blood volume, pH, directly involved in maintaining homeostasis, typically regulated by negative feedback loops. Meaning that whatever they correct, once they've corrected it, they stop. Like they'll detect an imbalance, and then once the imbalance is corrected, then it stops trying to correct, instead of it kind of running away with itself, which would be a positive feedback loop. The exteroreceptors, things that detect stuff outside the body, smell, taste, vision, hearing, equilibrium, that sort of thing. Mostly just lets the central nervous system know what's going on around you. So different types, you have chemoreceptors. So taste and smell would be external, blood pH internal. If you were tasting for the pH of somebody else's blood, I'd recommend that you see a psychiatrist. Photoreceptors respond to light, so that'll be external. Mechanoreceptors, hearing, gravity, body motion, position. Hearing and gravity, external, and then motion and body position are internal. We can kind of get a feel for where gravity is pulling us with our limbs and their relationship to our body and how they're being pulled one way or the other, but most of that is done with the semilunar canals in the ear. Thermoreceptors stimulated by a change in temperature. So hypothalamus, internal, regulates your body temperature. Skin, external. You have different, or just basically know this table and you're good as far as that's concerned. So how sensation occurs. You detect something in the external environment, and then you, so what that means is, something changes, such as pressure on fingertips, light in the eye, something like that. Something in the external environment happens that is of note for your senses to become aware of. Then that information, which is known as sensation, is sent down the nerve impulses and sent to the brain, and then perception occurs when you become consciously aware of it. As we've talked about before with like reflex arcs, you can move before you have perceived something. So a nice little summary of that. Let's see. Yeah, so this is a little redundant. We've already said all of this, but we'll go through it again here. So we're aware of reflex action when sensory information reaches the brain, but see, we've already done the reflex, so, brain integrates this information with other information received from sensory receptors. Some sensory receptors are free nerve endings, like sense of touch, or some of the senses of touch and pain and pressure in your skin. Others are specialized cells with neurons, so like the cones and rods in your eyes, for instance. And then the plasma membrane of a sensory receptor contains the proteins that react to the stimulus. Secondary transduction, energy from a chemical or physical stimulus is converted into an electrical signal, like a nerve impulse. The stronger the stimulus, the more frequent the action potential, and the sensation that results is dependent on the part of the brain receiving the nerve impulse. There is such a thing as synesthesia, where this sensation does not go to the correct part of the brain. So such people can do things as odd as smell colors and taste sounds. It's kind of interesting. Such people do actually tend to have much stronger memories than average because there's so much more sensory data coming in. You might not be able to remember off the top of your head what that word was that you needed to fill in that space in the vocabulary sheet, but it tasted like strawberries, and that kind of leads you in the right direction. It's like, oh yeah, that was it. So, it's bizarre, but potentially very useful. Integration occurs before the sensory receptors initiate the nerve impulse. Basically, it sums up everything that's coming into the nerve, like we talked about in the previous chapter, with that integration of the inhibitory and excitatory signals, forming the gestalt of the state that the nerve is in. Same kind of idea here. Sensory adaptation, a decrease in response to a stimulus to the point where you're not consciously aware of it. For instance, you may not have been aware of the way that your tongue is sitting in your mouth and how it's completely uncomfortable no matter which way you put it. It just keeps bumping into your teeth, or your shirt being draped over your shoulders and the collar around your neck. It's just quite irritating. It's itchy. Probably weren't even aware of that until I said anything, because you had already become adapted to it and you weren't consciously aware of it. So 2 ways that happens: the sensory receptors have stopped sending the impulse, or your thalamus has filtered it out. In the case of the 2 things I just mentioned, it's probably more that the thalamus is filtering out that information. Somatic senses are those that are associated with skin, muscles, joints, and viscera. So your 3 types are somatosensory, or 3 types of somatic sensory receptors are proprioceptors, and that tells you, where your bits are in relation to other bits. Cutaneous receptors, that's what's on the surface of your skin, pressure, hot, cold, that sort of thing. And then pain receptors, that pain has its own pathway. And as much as people dislike pain, it is massively useful to have. Proprioceptors are mechanical receptors involved in reflex action, help to maintain muscle tone. Muscle spindles increase the degree of the muscle contraction. Golgi tendon organs decrease the degree of muscle contraction. So these 2 are kind of going back and forth. Like, the muscle needs to have a certain amount of tension, but not too much that you're stressing the ligaments. So, when they kind of regulate how much you're supposed to use, and the result is you maintain proper length and tension of the muscle, which results in having muscle tone. So a little something like this. Get your little tendon or Golgi tendon organ right here, and it's stretched when the tendon is stretched, and it doesn't like that. So it tells the muscle, hey, lighten up, dude. And then you've got the muscle fiber here, and it wants to, be ready to act. So it needs a certain amount of tension in it to maintain, your posture and whatnot. So it, tightens up. So they're working in conjunction here with each other to maintain, whatever position is that you need. Cutaneous receptors continued. So we have you have your cutaneous and subcutaneous layers of your skin, your dermal and epidermal. I'm sorry, dermis and epidermis, and they have receptors in both. So myxin corpuscles, cross end bulbs, or Krauss end bulbs, however you pronounce that, in your fingertips, lips, palm, penis, and clitoris. The Merkel discs are junctions of the epidermis and dermis, are located at the junctions. And then the root hair plexus is a free nerve ending at the base of the follicles. That's how you can feel something when the hair is touched, because the nerve ending is stimulated. Also why it hurts when you have hair ripped out. Pressure receptors, so Paucian corpuscles, which are little onion-shaped, and they're deep on the dermis. Rufian endings, encapsulated receptors with complex nerve networks. Temperature receptors are free nerve endings. Some respond to hot, or to cold, which are more numerous, and some respond to cold. I said that backwards twice. Some respond to the cold, and they're more numerous, and some respond to warmth. That's why cold tends to feel more intense on people, and we can take heat easier. I'm kind of the other way around, just because of the way that I'm built. I have I'm rather densely constructed, so I overheat easy, and it needs to be fairly cool before I'm comfortable. But that changes as you age, by the way. The older you get, you lose a lot of subcutaneous fat, and you're less able to regulate your heat, which is why older people are always cold. So we've got our different cutaneous endings in the skin here. Just basically, this is a diagram of previous paragraph, so don't get too wrapped up in it. Pain receptors, free nerve endings, nociceptors. Know that nociceptor is for pain. Stimulated by chemicals released by damaged tissue and alert us to possible danger. So, without this, it would be very easy to, incur many small injuries throughout the day and never notice it, which would drastically increase your chances of getting an infection and dying. Or maybe you overexerted yourself and tore something. Well, if you're not aware of the pain caused by that, you're going to keep doing it, and you will destroy whatever that poor muscle or ligament or tendon is, or even sometimes, your viscera. So pain is incredibly helpful to survival and alerting people to danger, because, maybe you reach in, the stove's too hot, or you find a thorn bush, something. Or, a tiger just helped you lose 5 pounds off your rear end. Bonus points if you get that reference. Referred pain, actually I think it was a line, but bonus points if you get the reference. Referred pain, some areas stimulation is, or in some areas stimulation of an internal pain receptor is perceived as pain on the skin, because they follow, like, the same nerve track runs back to the central nervous system. So sometimes what they're saying here, the impulses from the internal pain, synapses in the cord with neurons receiving from the impulse from the skin. So same nerve track coming back. So it gets confused as to where it's coming from. So if you have damage to your heart, then you tend to feel pain in your left arm and shoulder. People coming into the ER complaining of chest pain probably have heartburn. Unless they also throw in shortness of breath with that, then maybe they're having a heart attack. But if they come in and they've got shortness of breath and their left arm is numb or in a lot of pain, then they're having a heart attack 9 times out of 10. Taste and smell, chemical senses, because the receptors are sensitive to molecules in the food we eat and the air we breathe. So, fun fact, if you can smell something, that means molecular particles of it are getting into the air and finding their way to bind to your nervous system. So, yeah. Taste cells and olfactory cells are classified as chemoreceptors. Sense of taste primarily comes from your taste buds, and you've got all these little chromatophores located mostly on the tongue, and they walk along the walls of the papillae. You have some in your palate, pharynx, and epiglottis, which is why sometimes if you breathe something in, you can taste it as much as smell it. So, yeah. Primarily we taste salty, sour, bitter, sweet, and umami, which is glutamic acid, or is present in MSG. Umami is just supposed to be like a savory kind of flavor, kind of like the same way that a savory sort of like meaty kind of flavor. Taste buds open at taste pores, surrounded by supporting cells and the taste cells. They have little microvilli with receptors to give them more surface area. And then the gustatory cortex interprets that as a particular taste. It kind of takes the gestalt of what all the buds are telling it and creates the flavor of something through the means of a weighted average. Which is why you can take something that tastes absolutely foul and cover it in sauce, and suddenly it's not so bad. Yeah, taste bud clusters. Got your tonsils. Anywho. Sense of smell. So the majority of what we taste is actually what we smell. So when you have a cold or a flu or something and your nose is stopped up and all of your olfactory nerves are covered in mucus, you're not really getting much in the way of taste. So Yeah. Basically, you have their chemoreceptors and or their modified nerve endings located high in the nasal cavity, and they have this little tuft of olfactory cilia with receptors for odor molecules. Each olfactory cell has only one of about 1,000 different types of receptor proteins, and each of those is a unique, um, or it sends a unique signal to the brain when it binds to its thing. So the brain's like, oh, okay, this nerve has bound, it's telling me we're detecting this. A single odor is composed of many different molecules which activate a characteristic combination of the receptor proteins, and so the odor's signal is what's interpreted by the brain. And so humans are a little funny. Um, I know everyone likes to go on about pheromones and everything, but for humans, there has not been a pheromone discovered that has any real effect on us in terms of, uh, estrus or rut or anything like that, like you would see in other mammals. Um, they have done experiments where they have noted that when you're smelling sweat and body odor from people, if it is for males, if a female is during the peak of her ovulatory cycle, then she smells more attractive. Um, and for females, males that have high levels of testosterone tend to smell better. Uh, but we don't know why. the The guess is that where they're all we can really smell is testosterone and estrogen, and having a preference for high testosterone and estrogen means that you're more likely to produce healthy offspring, or that a particular encounter is more likely to lead to offspring. Uh, but for humans, most of what we smell and our interpretations of it are based entirely on experience. So, what smells foul and ungodly to one person can smell like nostalgia and safety in childhood to another. That's why some people like absolutely love the smell of yeast, and some people it's just smells like foul, musty garbage. Uh, I was I spent some time as a child down the street from a bakery, and I love the smell of yeast. It meant that there were going to be fresh, hot honey buns shortly. Uh, but most people, when they first smell yeast, it smells terrible. So, a little bit of weirdness. Anyway, so the way this works, you got your olfactory bulb, which is an extension of your brain, just kind of laying up here on the inside of your skull. And it has little neurons that go down from it, and they end in these little uh, olfactory cilia. And, random scent molecules are wafting by and they bind to their receptor, and that sends a signal back to the brain, which is this right here is exactly how Naegleria fowleri, which is the brain-eating amoeba found in lake bottoms in several states, including Tennessee, in the mud and the water, which can be disturbed, particularly in the summer. People are, running around with boats and jumping into the water and everything. So if you get water in your nose, be very cautious. If you start developing meningitis-like symptoms, get to the hospital immediately and request that they do a wet prep of your cerebral spinal fluid, because that's about the only way to detect the organism. But anyway, so it gets on this olfactory receptor here and eats its way up into your brain via the olfactory bulb. Uh, sense of vision requires the work of the eyes and the brain. Believed to be at least a third of the cerebral cortex is required to process that information, which is why we kind of tend to focus like one thing at a time. We just don't have the computing power or the processing power in this meat hardware in between our ears to process everything in HD around us at any given time. So the eye is an oblong sphere, about 2.5 centimeters in diameter. You got your three layers: so sclera, outer layer, which also contains the cornea, which is made of transparent collagen, and the conjunctivae. It covers the sclera. Then you have your choroid, middle layer, darkly pigmented, vascular. To Towards the front, it becomes the donut-shaped iris, which is composed of smooth muscle to control the size of the pupil. Then behind the iris, it thickens into the ciliary bodies, and they contain ciliary muscles, which control the shape of the lens for near and far vision. And the lens is divided into two compartments. You have anterior, which is full of, or which is in front of the lens and is filled with aqueous humor, so clear watery fluid. And then the posterior compartment behind the lens has vitreous fluid in it. Retina is your third layer, located in the posterior compartment, filled with a clear, gelatinous, vitreous humor. Contains photoreceptors, your rods and cones, and has a special region called the fovea centralis, where the cones are densely packed. That's the part, like when you turn your eyes to look at something and you focus on one specific thing, basically you're pointing the fovea centralis at it, because that's where you've got the most definition and where the majority of your color vision comes from. Most of your periphery is actually in black and white, but as you'll see in the lab, the brain lies about what you can see all the time, including filling in your blind spots and adding color to the periphery based on what it expects to be there and that sort of thing. Optic nerve takes impulses from the visual cortex and is formed by the sensory nerves from the retina. So the lens focuses the image onto the retina, starts with the cornea and continues as rays pass through the lens and the humors. Visual accommodation, so basically just changing the shape of the lens depending on what you need. So for things that are the image can round itself up to bring images in focus with the retina. The shape is controlled by ciliary muscles, and as you age, you lose elasticity, so then you'll become presbyopic, if I'm remembering correctly. Let's see. Yeah, so just know this table, and that's all the details to remember for that. Rod cells, with their pigment is rhodopsin, which is incredibly sensitive to light. And the thing is rhodopsin breaks down in the presence of light. So it will, like the rods become less sensitive the more light is around, which is why if you've been in the dark for a few minutes, you can see better in the dark than when you started. And then if someone suddenly shines a light, then you are or turns the light on, then you're blinded temporarily till the rhodopsin breaks down. Cone cells, located in the fovea centralis, require bright light in order to be activated, and their thing is fine detail and color perception. You've got 3 different kinds, green, blue, and red, and different combinations are stimulated to produce different colors. So notice we can't actually see yellow or magenta. Magenta does not exist. There is no wavelength of light associated with it. It is something that our brains have fabricated because it thinks it's a logical thing to include in the color spectrum. So yeah, that's fun. The other trick here is having these cones are coded for by your X chromosome, which is why it's very unlikely for a female to be colorblind, because you would have to have 2 defective X chromosomes to be colorblind. And also there's slight variations within these, and that's the average woman can actually see a broader range of colors than the average male. Sometimes, in very rare cases, up to a million or up to 2 million more colors than the average male can see, which to me is biological evidence for why women care more about matching colors and coordinating things than males do. Because for males, it's all about the same, really. But for them, for females, no, those really are 2 different shades of blue, and the one does look hideous. So roll with it, guys. So yeah, just a visual representation here of what your different receptors look like. It's a function of the retina. You got 3 layers of neurons. Closest to the choroid contains rods and cones. Middle layer is composed of bipolar cells, and then the inner layer is composed of ganglion cells, which are sensory fibers that go back to form the optic nerve. You have yeah, it's just a visual representation. Your blind spot, like we mentioned before, is where the optic nerve attaches, and you've got your blood vessels coming into the eye as well. Layers of your retina. Again, visual representation of that. So rod and cone cells synapse with bipolar cells, which synapse with ganglions, and then the axons become the optic nerve. Sensitivity of cones and rods is due to how directly they connect to ganglion cells. So as many as 150 rods may synapse on the same ganglion. So if you've got 150 different things that can be like that dot, that pixel there forming your image, it's not going to have as high a definition when you have something like your cones, which form a one-to-one connection. So you can create a far more detailed picture with the cones than you can with the rods. Blind spot we already talked about. So visual pathway to the brain, you have information from the retina, which is then delivered to the visual cortex via the optic nerve, which goes back, the optic nerve travels back to the optic chiasm, which crisscrosses. So the information from the right eye is being processed by the left side of the brain and vice versa. Yeah. So then once you get to the optic tract, it goes around to the hypothalamus, and most of the fibers then are around the hypothalamus, and then the fibers synapse with the nuclei in the thalamus. And then the thalamus, the axons from the thalamus form optic radiations that carry impulses to the visual areas of the brain, back at the occipital lobe, among others. And then the right and left visual fields must communicate for us to see the whole thing. So, a little something like this. Hearing has 2 functions. So, the ear has 2 functions: hearing and balance. So, well, and kind of 3, because it also helps you orientate yourself to gravity, but that's more or less part of balance. So the sensory receptors for both of these are located in the inner ear, and they consist of hairs with stereocilia, which are basically like little long macrovilli that are sensitive to mechanical stimulation, mechanophores. So 3 divisions of the ear. You've got the outer ear, which collects and funnels sound to the auditory canal, which is lined with fine hairs and sweat glands, which produce modified sweat, which is earwax, basically. The middle ear begins at the tympanic membrane and ends in a bony wall with the openings called the oval and round windows. And between the tympanic membrane and the oval window are 3 bones, which are the ossicles, the malleus, incus, and stapes. The malleus looks like a hammer, the incus looks like an anvil, and the stapes looks like a stirrup. Which you then have from there the auditory tube that extends from the middle ear to the nasopharynx, permitting equalizing of air pressure, your eustachian tubes. The inner ear is the only part that's fluid filled, so you've got your semilunar canals, which help with equilibrium, your vestibule, also equilibrium, and then the cochlea, which is hearing. So the semilunar canals are the 3 like ring-shaped things, and the vestibule is like at the bottom of that, and the cochlea is the thing that looks like a snail shell in your ear. So something like this. This auditory tube is also called the eustachian tube. When you yawn, it opens this up more. That's so you can equalize pressure. That's why your ears will often pop when yawning if you have been traveling uphill or on a flight or something. Through the auditory and middle ear, you then begin the process of converting sound waves. So the tympanic membrane vibrates with the high and low pressure waves in the air, and then the vibrations are amplified across the ossicles, and that's transmitted to the stapes, which is on the oval window, which then vibrates and transmits that to the fluid inside the cochlea. And then you get to inside the cochlea, you've got your fluid-filled canals. The vestibular canal is filled with perilymph, the cochlear is endolymph, and your tympanic canal is again perilymph. The sense organs for hearing is called the organ of Corti. When you have hair cells that sit on the basal membrane with stereocilia embedded in this tectoral membrane. When the stapes strikes the oval window of the membrane, pressure waves move from the vestibular canal to the tympanic canal across the basilar membrane, which then moves up and down, and that agitates the stereocilia in the tectoral membrane, which then transmit nerve impulses through the cochlear nerve to the brain. When they reach the auditory cortex of the temporal lobe, they are interpreted as a sound. So each part of the organ of Corti is different to different wavelength frequencies or pitch. So near the base, like the start of the snail shell, basically, it's lower, responds to lower pitch. Oh, I'm sorry, I said that backwards. Near the tip, lower pitches, and near the base, higher pitches. So depending on which region of the basilar membrane vibrates, then you have a different part of the auditory complex that's stimulated, and you can interpret that as different sounds. Volume is a function of the amplitude of the waves, so loud noises cause the fluid to exert more pressure, and the basilar membrane vibrates more, and the brain interprets that increased stimulation as loud, or as being louder. A little something like that. So remember, again, your semilunar canals here, that's for balance, orientation, and your little snail shell cochlea, round window. That's inside here, you've got your vestibular, cochlear, and tympanic canals, and then you've got your tympanic membrane, or you've got your membranes here with the microcilia attached to them to send down to the cochlear nerve. So tectoral membrane, stereocilia, I said micro, I meant stereo, and then attached to the basal membrane. So sound comes through, vibrates this, bends these, sends a nerve impulse. Looks a little something like that. Rotational equilibrium. So you've got those three semilunar canals, so that you've got one orientated in each direction. So they're 90. They're all 90 degrees from each other. So you can have three-dimensional awareness in space. Each one has each semilunar canal has an enlarged base called the ampulla, which contains hair cells with more stereocilia embedded in the cupula. And as fluid flows in the canal, it bends the cupula. So it's basically they're like the rings sitting there that are hollow on the inside. And if you are changing direction in the plane associated with a particular canal, then the fluid will move. And since you've got them all rotated 90 degrees from each other, you can, depending on the degree of movement in all three of them, you can tell exactly which way you're moving relative to the pull of gravity. So you use all of that information in order to figure out where you are, and then you make postural corrections to stay upright and all of that. So gravitational equilibrium depends on the utriole and saccule. The utriole is sensitive to horizontal movements, the saccule is sensitive to vertical, and they both contain hair cells with stereocilia embedded in an otolithic membrane. Large central cilium called the kinocilium, calcium carbonate granules, the otoliths. rest on the otolithic membrane. So when you move in a vertical or horizontal plane, the otoliths are displaced and the otolithic membrane sags, bending the stereocilia. So something like this here. Well, this is in your semilunar canals right here with the fluid moving, bends the things around. And then here you've got those little otoliths that we're talking about. So disorders that affect the senses. So for taste and smell, usually around 60 you begin to lose your sense of smell. Some people are born without it, and that's referred to as anosmia. Other factors that can contribute to a decreased ability: upper respiratory infections, allergies, exposure to drugs or chemicals, in particular tobacco smoke, brain trauma, color blindness, and problems with the visual field. You've got color blindness and problems with visual focus are the two common abnormalities of the eye. More serious disorders result in blindness. So color blindness, complete color blindness is rare. You're typically deficient in one or two types of cones, but if you're completely colorblind, you don't have any cones. So red-green colorblind, most common, X-linked recessive, 5 to 8% of males, 0.5% of females. You've got this little, this one would test for red-green. So you've got the numbers here in green with red and yellow background. Visual focus, so if you're nearsighted, you can see things up close better than distant ones. It's because the eye's been elongated and it changes the point of focus on the retina. Can be corrected with a concave lens, your glasses. Farsightedness, exactly the opposite. So, corrected convex lens, the eye is shortened. Astigmatism, you have your cornea or your lens in your eye is uneven. Don't think that the cornea is the lens; those are two separate things. And it ends up producing a fuzzy image, or like parts of it are wavy or distorted, because it's unevenly focusing. So you have to have lenses ground specifically to compensate for that. So this is how glasses work here, basically. It changes the focal point. So here, like the light's focusing there, so past that point the rays diverge and they're not focused anymore, so it's a blurry image. Put the glasses on and it shifts the focal point back. Similar thing here for like uneven cornea. So it's just, the lens has to be specific to correct where the problem is, so it's thinner here where it's in order to help focus it. Retinal disorders, so diabetic retinopathy. Basically, if you are diabetic, and you're not controlling it, then you have very high blood sugar. If you're a type 1 diabetic, that's autoimmune, probably. We don't entirely know, but the current theory is that your beta cells in your pancreas were collateral damage because you got some infection or something that was similar. The antigens on the surfaces of it were similar to the ones on your beta cells, so your immune system wiped them both out. Type 2 means that, basically, and this is a massive oversimplification, but basically you produce enough insulin for a person of a particular size, and once you are X amount over that size, you no longer produce enough insulin to go around, and that can lead to insulin resistance because you're pumping out tons and tons of insulin, and your body likes to try to compensate to stay at equilibrium, so you can actually reduce the number of insulin-sensitive pumps on the surface of your cells because they think they're being overstimulated because you're cranking out all this extra insulin trying to keep up, and so then you become insulin resistant and it drives your blood sugar even higher, or you can burn out your pancreas and you'll end up producing less insulin overall. But when that happens and you have very high blood sugar, the first thing that is usually destroyed is your nerve endings, and it's more severe in the periphery than towards the median of the body. And over time, that leads to diabetic neuropathy. So you have like numbness, like in the feet and hands and whatnot. This also, the high blood sugar can compromise your immune system, and that can lead to a lot of kidney damage because you can't reabsorb that much sugar. You're constantly passing it through your kidneys. So that's can lead to diabetic nephropathy. And then you can also damage all of your small blood vessels. In this case, for retinopathy, it's the capillaries in the retina. And that can lead to blindness. Because if you kill the retina, you can't see anything. Macular degeneration, your cones are destroyed because the thickened choroid vessels no longer function. Retinal detachment, you have following trauma, then the retina can be torn or separated from the choroid. This can also happen with some infections. Glaucoma, you have fluid buildup in the eye because the drainage system fails, which leads to an increased pressure, and that can damage nerves with the periphery due to the excess pressure. Cataracts are cloudy spots on the lens, which will eventually cover the entire lens. So risk factors: exposure to UV, diabetes, heavy alcohol consumption, smoking. If you have blue eyes, there is no pigment for blue eyes. It's actually the same pigment that's in brown eyes. The thing is, you just have a lot less. So the light is being bent and distorted to look blue instead of reflecting back the color of the pigment. Just like the sky is not really blue. It's the light is being bent to make it look blue. So, Rayleigh scattering, I think it is. Google Rayleigh scattering to get more information on that if you want. So if you're blue-eyed, you need to be wearing sunglasses or transition lenses. Now the upside to that is blue eyes can see better in the dark. So plus they also blue eyes confer a massive selective advantage because fathers with blue eyes typically know that their blue-eyed offspring are in fact theirs, or at least historically speaking, that's kind of what led to the massive upswing in the spread of blue eyes through the population, because then males are significant of all species, but especially humans, are significantly more likely to actually stick around and provide resources and parental input if they know that the offspring are theirs. So that helped. It was some mutation, thousands of years ago in Europe, was when blue eyes first originated, and that became dominant in that area. So as a result, you would expect that males would have a preference for blue-eyed females, or blue-eyed males have a preference for blue-eyed females, and they do. Females, on the other hand, have no such preference. They don't seem to care too much about the color of their significant other's eyes. But tangent aside, you have blue eyes, wear sunglasses. So that's cataracts. Now, about the only thing you can do with that is you make an incision right along the edge of the cornea, and you bust this up with a sonic device and pull it out through the incision. You gotta make sure you got all the bits and pieces loose too. I think maybe you can actually cut it with scissors or something too. It just kind of depends on the circumstances. So you get all the pieces out. Now, without a lens, you can't really focus too well, but that's better than everything being a blur. Or everything just, this eye right now, about the only thing it can tell is the difference between light and dark. You pull that lens out and you get color and rough shapes. So eventually we'll be able to just swap it out with a new one. I think there are actually like artificial lenses that can be replaced in some cases, but not all. Hearing loss can develop either suddenly or gradually. 1 in 3 people over the age of 60 have hearing loss. Psychologically speaking, the loss of the ability to hear is actually far more devastating to people than the ability to see. They feel isolated and alone when they can't hear. Blindness, you feel kind of weak and helpless, but you still are very aware of everything going on around you. You just kind of have to retool and reorientate yourself, and you're still pretty aware and with it, in touch with what's going on. If you're blind, you can only see what's right in front of you. Or I'm sorry, if you're deaf, you're only really aware of what's going on right in front of you. They can't hear people. They're constantly just in a silent void, and they feel isolated and alone. So that's fun. Um, so 1 in 3 people over 60 have hearing loss. The middle ear is subject to infections that can lead to impairment if not treated properly. First sign is usually difficulty understanding conversations when there's background noise. Some people develop hearing problems as early as 20, and the motility of your ossicles will decrease with age, and that also will decrease your ability to hear. Sudden deafness usually occurs in a single ear at a time, usually caused by infections, trauma, side effects of some drugs. Flashbang going off right next to your head will blow out your eardrums and cause significant damage to your ear. Sometimes it'll heal, sometimes it won't. Some people are born deaf. If you get German measles or mumps during pregnancy, then that can cause birth defects in the child, leading to them being born deaf, or you can have genetic disorders. Vertigo is a feeling that the person or the environment is moving when no motion is occurring. You can kind of get a sense of that if you spin in a circle for long enough and then just stop. But so imagine that, but all the time, or just spontaneously triggering, depending on how bad you've got it. And it can be caused by problems in the brain or the ear. So the ear is the thing that is supposed to be collecting this information, and the brain's the thing that's supposed to be interpreting it. So if either one of them are messed up, then you get false signals. Benign positional vertigo due to particles in the simular canals. So you just got gunk in there, crystals or something that have formed, and depending on how you turn, it can give you it overstimulates or whatever, and you get a false sense of things moving. Meningeals disease caused by an increased fluid volume in the inner ear. You got vertigo, feelings of fullness in the ears, tinnitus, and hearing loss. And that is everything for Chapter 18. If you've got any questions, comments, concerns, let me know, and I'll see you next time.